When a fruit fly is infected with a lethal bacterium, its body immediately initiates a microscopic war to contain the invader. We can't see the strategy of this defense just by looking at DNA. While the DNA holds the cell's entire library of instructions, it is static. It remains the same whether the fly is healthy or dying, meaning it cannot reveal what the immune system is actually doing in the heat of the moment. To see the active response, we look for RNA. When an immunity gene switches on, the DNA unzips to produce a messenger RNA strand, a mobile copy of the instructions that travels out of the nucleus to begin building defensive proteins. By capturing and reading these RNA messengers during the exact moment of infection, we can map the specific genes the fly chooses to deploy against a live threat. Tracking this response requires a controlled comparison. We must process a group of infected flies alongside a healthy control group to ensure we isolate the changes caused specifically by the bacteria. Researchers grind the fly tissue to release the internal components. Through a series of chemical washes, they physically isolate the raw nucleic acids, stripping away the cell's structure to leave only the RNA behind. However, modern sequencing machines have a physical limitation. They cannot read whole genomes or even long RNA strands in one go. We bypass this through library preparation. The long RNA molecules are shattered into millions of tiny fragments. Artificial adapters, or primers, are then attached to the ends of each piece, so the sequencing machine has a consistent chemical handle to grip. The biological sample is now effectively a digitized chemical library, ready to be read by a computer. Inside a next-generation sequencing machine, these fragments are converted into digital code. The device outputs massive text files, known as FASTQ files, containing millions of strings of the letters A, C, G, and T. The scale of this data is massive. A single run generates millions of disconnected short reads, which on their own carry no immediate biological meaning. This raw data often contains experimental errors, similar to ink splashes on a magazine page. Before analysis can begin, algorithms must trim away the artificial adapters and discard any low-quality reads that could skew the results. At this stage, the biological sample is a disordered collection of segments. To make it useful, we need a computational method to organize these fragments back into their original structure. We achieve this through genome alignment. Algorithms compare fragments against the reference genome, locking them into matching positions. As fragments are mapped into structured BAM files, reads stack up like vertical pillars over specific genes. During gene counting, the algorithm measures the depth of these stacks, transforming visual piles into a structured matrix of numerical values representing precise mRNA amounts. The algorithms have successfully converted chaotic digital noise into a quantifiable count matrix, grounding the digital data back in biological reality. Raw numbers can be misleading due to variations in sample size or total RNA content. We use mathematical normalization to adjust the matrix, ensuring a fair comparison between the infected and healthy samples. To verify the data, we use principal component analysis to cluster the samples. This confirms that the genetic behavior of the infected flies is distinctly different from the healthy ones, proving the signal we're seeing is biological, not just random variation. We visualize this matrix with a heat map. This grid shows the exact contrast between the groups, with bright red regions highlighting the genes that were switched on to fight the infection, and deep blue regions showing the genes that were suppressed. Statistical filters have now stripped away the background noise of the entire genome, isolating the specific genetic signal responding to the bacteria. This leads to differential expression analysis, where we identify the specific immunity genes, such as NF-kappa B, that spiked to defend the fly. Analysis of these genomic sequences reveals that fruit flies and humans share significant genetic similarities, including genes linked to complex diseases. By mapping these identified fly genes to broader functional networks, we observe the same metabolic and regulatory pathways operating in our own bodies. These findings are uploaded to public repositories for the global scientific community and synthesized into published research. This pipeline translates raw tissue into a targeted list of metabolic leads. By isolating these specific genetic responses in the lab, researchers can identify the functional units of disease and begin developing more effective modes of treatment.